Hello everyone, this is Dr. P. Mary Anupama, Head of the Department and Assistant Professor of the Department of Biochemistry, St. Joseph's College for Women. Today we'll be studying the topic Gluconeogenesis. The following topics will be covered. So these include location of gluconeogenesis, the importance, and the precursors of gluconeogenesis, followed by the reactions that take place, then gluconeogenesis from amino acids, glycerol, propionate, and even from lactate, which includes the Cori cycle, then glucose and alanine cycle. This will be followed by energetics of gluconeogenesis and then its regulation. Now, what is gluconeogenesis? It is the synthesis of glucose from non-carbohydrate compounds. So these include, like we have lactate, pyruvate, and some glucogenic amino acids are there, propionate as well as glycerol. All these, they undergo some reactions which we call them as the gluconeogenesis and finally result in the formation of glucose. Location of gluconeogenesis, where it takes place. The major organ where gluconeogenesis takes place is the liver. Around 1 kg of glucose it is synthesized every day by the liver. Next comes the kidney matrix. This also significantly contributes to gluconeogenesis, and this occurs mainly when a person is doing prolonged fasting or acidosis, under acidosis. Other tissues are also there which do it, like the small intestines and the brain. They do minimal amount. And coming to cellularly, gluconeogenesis occurs in both mitochondria as well as cytosol, with some steps that particularly take place in the mitochondria, like the conversion of pyruvate to oxaloacetate, while the rest of the steps they take place in the cytosol. So here liver is the major organ that takes part in gluconeogenesis. Next is the kidney matrix. Importance of gluconeogenesis. First one is it is involved in maintaining blood glucose levels, mainly during fasting and starvation. Because human brain, it requires around 120 grams of glucose per day. So out of 160 grams of glucose which is required for the body, 120 grams goes to the brain. So that's why this is the first important one, maintaining blood glucose levels. Next one is supplying glucose to glucose dependent tissues, which include our brain as well as red blood cells. Next one is preventing hypoglycemia. So during when a person is doing prolonged exercise or when a person is doing this caloric restriction diet, under such conditions, the blood glucose level comes down. So gluconeogenesis prevents this from happening. Next is managing diabetes. It provides glucose in conditions of diabetes, and it does it where insulin deficiency is also there, or the resistance. It impairs normal glucose uptake. Next one is detoxifying. So the pathway also, it serves as a critical function in detoxification of the lactate via the Cori cycle. As well as you have some amino acids, you have the glucose-alanine cycle, so wherein it clears the amino acids through glucose-alanine cycle. So all this contributes to the importance of gluconeogenesis. Precursors of gluconeogenesis. There are several non-carbohydrate precursors that contribute to this gluconeogenic pathways. The first one is the lactate. So lactate, it is produced during anaerobic glycolysis in the muscles and in the RBC also. So this lactate, it is transported to the liver where it is converted back to glucose via the Cori cycle. Next is glycerol. So glycerol, it is derived from the hydrolysis of triglycerides in the adipose tissue. So the glycerol is first converted to dihydroxyacetone phosphate in the liver. Next is the glucogenic amino acids. So you have alanine mainly, It plays important role via the glucose-alanine cycle. Next we have propionate. It is an important precursor seen in the ruminants, and it is formed from the breakdown of odd chain fatty acids and also some amino acids. Next is TCA cycle intermediates. These include malate and oxaloacetate, which are derived from amino acid metabolism. All these they act as precursors or the feed for gluconeogenic pathways. So here again, the glycerol, which is derived from the triglycerides of adipose tissue, it is transported to liver and then converted to dihydroxyhistone phosphate, while lactate from the muscles or RBC, it is transported to liver, which is converted to glucose by Cori cycle. You also have glucogenic amino acids like the alanine. They go through this glucose-alanine cycle and get converted to glucose, while propionate as well as the TCA cycle intermediates, they also undergo gluconeogenesis and then result in the production of glucose. Now coming to reactions of gluconeogenesis. So there are several reactions which are in common with the glycolysis. So gluconeogenesis, it shares many reversible reactions with the glycolysis. While there are also 3 steps of glycolytic cycle which are essentially irreversible, while the other 7 they are common for both glycolysis as well as gluconeogenesis. So the 3 irreversible steps of glycolysis, they are catalyzed primarily by these 3 enzymes, which are the hexokinase, phosphofructokinase, and then pyruvate kinase. These 3 stages, these are bypassed bypassed by alternative enzymes of the gluconeogenesis. The first step or the first reaction of gluconeogenesis is conversion of pyruvate to phosphoenol pyruvate. This step actually it bypasses the pyruvate kinase reaction in glycolysis, and it occurs in 2 steps. First one is the pyruvate is converted to oxaloacetate. So this reaction, it is catalyzed by the enzyme pyruvate carboxylase, which is present in the mitochondria. And this reaction, it requires biotin as well as ATP. Next, the oxaloacetate produced, it is converted to PEP, that is phosphoenol pyruvate. So this conversion, it is catalyzed by the enzyme PEP carboxykinase. PEP carboxykinase. And this reaction, it takes place both in the mitochondria as well as in the cytosol, and it requires the presence of GTP. So this oxaloacetate, it as such cannot cross the mitochondrial membrane. So often what happens is the oxaloacetate is converted to malate or aspartate, and then it is transported to the cytosol. So there it is converted to PEP. Okay. So here once it is transported, again it is reconverted back to oxaloacetate in the cytosol, and then PEP is formed. So that is how this second reaction takes place in the mitochondria as well as in the cytosol. Second step is conversion of fructose 1,6 bisphosphate to fructose 6-phosphate. This reaction, it bypasses the phosphofructose kinase 1 step in glycolysis, and it is catalyzed by fructose 1,6 bisphosphatase. So this is a regulatory enzyme also. And this step, it is irreversible and it is regulated by energy status and even hormonal signals. Third step is conversion of glucose 6-phosphate to glucose. So the enzyme glucose 6-phosphatase, it catalyzes this final step, which is involving conversion of glucose 6-phosphate into free glucose that can be released into the bloodstream. So this enzyme as such, it is located in the endoplasmic reticulum and is present only in gluconeogenic tissues, which include liver and kidneys. So here you can see the glucose 6-phosphate, it is converted by the enzyme glucose 6-phosphatase, and then finally it is converted to glucose, and the glucose is released into the bloodstream. The overall summary of gluconeogenesis for the conversions of pyruvate to glucose is shown here. So we have 2 molecules of pyruvate plus 4 moles of ATP, 2 GTP, 2 NADH plus and 6 water molecules will result in the formation of 1 molecule of glucose. We all know that in glycolysis, 1 molecule of glucose will lead to pyruvate, while in the reverse reaction, 2 pyruvate are resulting in the formation of 1 glucose molecules. So here you get 2 NAD plus, 4 ADP, 2 GDP plus 6 Pi and 6 protons. This is the overall summary of gluconeogenesis while pyruvate is getting converted to glucose. So this is the overall pathway of the reactions, while this is the glycolysis pathway which we all know. The reverse of it is the gluconeogenesis. You can see that all the enzymes, they are common for 7 steps, while from here the enzymes are varying, which we have discussed till now. So gluconeogenesis starts from pyruvate to formation of glucose. The enzymes are common, while here the enzymes, we have studied them just now, which are different. Now let us see gluconeogenesis from amino acids. The carbon skeleton of glucogenic amino acids, like most of the amino acids except leucine and lysine, they result in the formation of pyruvate, or they may also result in the formation of intermediates of acetic acid cycle. So these ultimately result in the synthesis of glucose. So the glucogenic amino acids carbon skeleton, it is used either for production of pyruvate or acetic acid cycle intermediates, which will lead to glucose synthesis. Gluconeogenesis from glycerol. So we know that glycerol, it is released mainly in the adipose tissues when the fats are hydrolyzed, like we have this triacyl glycerols. Fats are triacyl glycerols. When they are hydrolyzed, they result in the production of glycerol. So glycerol is released. So we have the enzyme glycerokinase, which is found in liver, kidney, while it is absent in the adipose tissues. So this glycerokinase, it activates glycerol, and glycerol gets converted to glycerol 3-phosphate. Later on, it is converted to dihydroxyacetone phosphate by the enzyme glycerol 3-phosphate dehydrogenase. Then the dihydroxyacetone phosphate, which is the intermediate, we know that DHAP is the intermediate of glycolysis. So from there the steps will proceed and ultimately glucose production takes place. So this is how from glycerol glucose is produced. Then gluconeogenesis from propionate. So when the odd chain fatty acids, while they are being oxidized, and in the breakdown of some amino acids, like we have our methionine as well as isoleucine, they all result in the formation of propionyl CoA. So this propionyl CoA, it is acted upon by propionyl CoA carboxylase in the presence of ATP as well as biotin, and that will get converted to methyl malonyl CoA. So the methyl malonyl CoA, it is further converted to succinyl CoA in the presence of the cofactors or coenzymes, and the succinyl CoA here enters the gluconeogenic pathways via the citric acid cycle. So that is how from propionyl CoA, that is from propionate, which is converted to propionyl CoA, you get glucose. So propionyl CoA is getting converted to methyl malonyl CoA by propionyl CoA carboxylase. And here methyl malonyl CoA is converted to succinyl CoA, and from the succinyl coenzyme A, you are getting glucose via citric acid cycle. Next is gluconeogenesis from lactate, which takes place through the Cori cycle. So we all know that lactate, it is produced by the skeletal muscles when they are very active, and it is one of the major precursor for gluconeogenesis, the lactate. So under aerobic conditions, we all know that pyruvate, it is initially converted to lactate by the enzyme lactate dehydrogenase. So pyruvate in the presence of NADH plus H plus, in the presence of lactate dehydrogenase, you get this lactate or lactic acid and NAD plus. So here lactate, it is a dead end in glycolysis, and it must be reconverted back to pyruvate, okay, for its further metabolism. So the very purpose of lactate production is to regenerate NADH so that glycolysis proceeds uninterrupted in skeletal muscles. So lactate or pyruvate produced in the muscles, they cannot be utilized for the synthesis of glucose due to the absence of the key enzymes of gluconeogenesis, that is the glucose-6 phosphatase and fructose 1,6 phosphatase. The lactate which is produced in the muscles, this is the muscle. So during strenuous exercise, you see that lactic acid, glucose is converted to lactic acid or lactate. And now this membrane, it is permeable. The muscle membrane is permeable to lactate. So why is muscle unable to do this gluconeogenesis? Is because of the absence of glucose-6 phosphatase and fructose 1,6 bisphosphatase. So since the plasma membrane is freely permeable to this lactate, lactate from skeletal muscles enters into the bloodstream. Okay, so here the lactate from here through the blood, it is carried to liver, where it is oxidized back to pyruvate. So this pyruvate which is produced from here, this is converted to glucose here itself. So once the glucose is produced via the gluconeogenesis, either it is converted to glycogen or again it enters into the bloodstream and then again it reaches the skeletal muscles. So this is like a cycle wherein glucose is converted to lactate, lactate is carried to the liver, and the liver converts lactate back to glucose, and the glucose is transported back to the muscles. So the cycle, it is involving synthesis of glucose in liver from skeletal muscles, lactate, and the reuse of glucose thus synthesized by the muscle for energy purpose. This is known as the Cori cycle. So here is the definition of Cori cycle. The cycle involving the synthesis of glucose in liver from skeletal muscle lactate and the reuse of glucose thus synthesized by the muscle for energy purpose is known as Cori cycle. Next is the glucose-alanine cycle. So there is a continuous transport of amino acids from muscle to liver, which mainly is seen during starvation. And here you have alanine, which dominates among the transported amino acids. It's the most common one which is transported. and it has been postulated that pyruvate in the skeletal muscles, it undergoes the transamination reaction, and finally pyruvate is converted to alanine. So from pyruvate, alanine amino acid is produced. So this alanine, it is again transported to the liver, where it can be used for gluconeogenesis. So what is happening is the alanine is getting converted to glucose in the liver via the glucose-alanine cycle. Let us understand the energetics of gluconeogenesis. So gluconeogenesis as such, it's a very energy consuming process. Here, the synthesis of one molecule of glucose, if you take pyruvate as a substrate and when you are trying to convert pyruvate to glucose, it requires around 4 ATP, 2 GTP and 2 NADH. So here the energy investment alone drives this gluconeogenic pathway, while it is the exact opposite of glycolysis. Here is the overall reaction of pyruvate conversion to glucose. 2 molecules of pyruvate, 4 ATP, 2 GTP, NADH, 2 H plus and 6 water molecules gives rise to 1 glucose molecules, 4 ADP, 2 GDP, 6 inorganic phosphate and 2 NAD plus. So this is a very high energy demanding pathway, and it is driven mainly based on energy availability. Now it is about the regulation of gluconeogenesis. So here regulation is mainly controlled by hormones, which we call it as the hormonal regulation, as well as allosteric mechanisms. And these ensure that glucose is produced only when it is required by the body. Coming to hormonal regulation, here you have insulin, glucagon as well as cortisols, which play important role. Insulin as such, it inhibits the gluconeogenesis, and it also decreases the expression of key enzymes like PEP carboxylase kinase and glucose-6 phosphatase, so thereby decreasing the expression of these enzymes and inhibiting gluconeogenesis. Next one is our glucagon. Glucagon does the antagonistic function as insulin. So here it stimulates gluconeogenesis, and this is done mainly when a person is fasting. And this particular hormone, it activates the expression of gluconeogenic enzymes, and it also increases the synthesis of cyclic AMP, thereby leading to the activation of the protein kinase A. Next is cortisol. Cortisol as such, it promotes gluconeogenesis. What it does is it increases the transcription of enzymes that are required for gluconeogenesis. Next is our allosteric regulation. Allosteric regulation of by pyruvate carboxylase, fructose 1,6 bisphosphatase and then glucose 6 phosphatase. Pyruvate carboxylase as such is activated by acetyl-CoA, and this indicates a high energy state. Then fructose 1,6 bisphosphate. So this is inhibited by AMP and also fructose 2,6 bisphosphate, while it is activated by citrate. Next is glucose 6 phosphatase. Its activity, it is mainly influenced by the presence of substrate. Substrate availability induces production or synthesis of this particular enzyme. So let us summarize what we have studied. We have seen where gluconeogenesis takes place, its importance, the precursors that can be used for gluconeogenesis, and we have dealt some reactions mainly from pyruvate, followed by how gluconeogenesis occurs from amino acids, glycerol, propionate, lactate. We dealt Cori cycle in detail, followed by glucose-alanine cycle. Then we have seen the energetics of the pathway and then regulation of gluconeogenesis. And these are the textbooks that are referred: The Principles of Biochemistry by Lenicher, Biochemistry by U. Satyanarayana. Then we have also referred Harper's Biochemistry. Thank you.